This is Chapter 3, the cells, or cells is the name of the chapter. It begins on page 37 of your book. The different cell variations. You can see has a list. The first one is nerve cells. That's these right here at the top. Okay, these have long extensions, and this is what allows these cells to transmit impulses to other areas of the body. Then you have a muscle cell. This allows for movement. You have your red blood cells, which are a type of cell that don't have a nucleus, and these are used to transfer oxygen, nutrients throughout the blood, and they can move throughout blood vessels. You have glands, which secrete hormones and other things like enzymes and sweat. And then white blood cells, which are used to help fight infection and protect the body from foreign invaders. The structure of the cell. Each cell has a specific structure, even though if it's a different type of cell, like a liver cell or a heart cell, they have similar structures. So we're talking about the nucleus. We talked about the plasma membrane. We also talked about these extensions of the cytoplasm, and we talked about the mitochondria. We'll review them in more depth. The plasma membrane. This is a cell, and this particular area is a little portion that has been magnified for the plasma membrane. The plasma membrane is made up of phospholipids. It's a phospholipid bilayer because it has two layers. You have the hydrophilic heads and you have the hydrophobic tails, which these are the two tails. You have two different layers. Okay, the hydrophobic or the water-fearing line up together. If you look on page 39, that's where you'll see the plasma membrane. In the purple box, it'll be describing the hydrophilic heads, which are water-loving, and the hydrophobic tails, which are water-fearing. Embedded in the plasma membrane, you have different things. In the peach-colored box, it talks about cholesterol. That is these red dots that will give the plasma membrane structure. It helps stiffen the plasma membrane as well as strengthen it. Then you have different kinds of proteins that are embedded in the plasma membrane. This first one, it talks about, it's in the purple colored box. It says most proteins pass all the way through the membrane and act as channels. So you can see this is a channel to allow different solutes or particles go through the plasma membrane. The middle of that purple box, you'll see that it says some proteins attached to the surface of the membrane. These serve as receptors for specific chemicals. So those are the ones where the chemicals have to be able to have a certain formulation to be able to connect with those receptors in order to enter the cell. And then the third one towards the right, you can see many proteins have carbohydrates, these are representing the carbohydrates attached to their outer surface. These are called glycoproteins. The glycoproteins act as markers to help the body distinguish its own cells from foreign invaders.
Structure of the plasma membrane. The plasma membrane is the outer boundary of a cell. It is composed mainly of a double layer of phospholipid molecules that are held together by chemical attractions. These phospholipid molecules have a polar hydrophilic water-loving head and two nonpolar hydrophobic water-fearing tails. The membrane is formed as the heads face out and the tails face in, forming a bilayer that acts like a fence, as you can see here. The inner part of the cell, called the cytosol, is here, and the outside of the cell is here. Cholesterol is another molecule that is embedded in the plasma membrane, and it helps strengthen and stabilize the membrane. Glycolipids are lipids with carbohydrates attached to them, and they are found on the outer portion of the membrane, so they're exposed to the outside of the cell or the interstitial fluid that surrounds cells. They help maintain the stability of the membrane and aid in cell recognition. Proteins are also dispersed throughout the membrane, and they have several key roles. There are two structural types of membrane proteins, integral proteins and peripheral proteins. Integral proteins are embedded in the membrane and extend across the membrane, with many being glycoproteins, which are proteins with carbohydrates attached. The carbohydrate portions of the glycoproteins and glycolipids help to form what is called the glycocalyx, which is a sugar-like coating on the outside of a cell's surface. Peripheral proteins are attached to the internal or external surface of the membrane, and many times they are attached to an integral protein. These membrane proteins have many important functions. Transport proteins regulate the movement of substances across the membrane. Some act as identity markers to communicate with other cells. Some are enzymes taking part in chemical reactions. Some act as anchoring sites, securing the cytoskeleton to the membrane, and some are part of cell-to-cell attachments. And that be the basics on the structure of the plasma membrane.
Okay, then we're talking about the nucleus. The nucleus is the most important part of the cell. On page 40, it says the central and most important part of the cell is the nucleus. The nucleus is the cell's control center, and this microscopic structure contains all the cell's genetic information. So the nucleus is where you're going to find your genetic information, and again, it's the control center of the cell. You have different organelles, which are inside the cell. These are kind of like, if you want to think of them as mini organs. And the one that we focused on is called mitochondria. Mitochondria, the other name for mitochondria, is referred to often as the cell's powerhouse. Mitochondria, it has two layers. It has the outer membrane and an inner membrane. The inner membrane folds back and forth across the interior, and the spaces between these enzymes is used to convert organic compounds into adenosine triphosphate. Remember, something that's organic contains carbon. So if you think of the carbon, something that has carbon in it being transferred into ATP. So ATP is energy, so it makes sense that the mitochondria is called the powerhouse because it's making the ATP.
Our body is made up of trillions of cells. They all require energy to function. This energy is created within our cells, in the mitochondria. Here, food is converted into chemical energy called ATP. ATP is released by the mitochondria so cells can use it. Mitochondria consist of two membranes, an outer membrane, separating it from the cytosol, and an inner membrane, surrounding the so-called matrix. The area between these membranes is called the intermembrane space. ATP is generated at the inner membrane of mitochondria by an efficient mechanism called oxidative phosphorylation, involving several membrane protein complexes. Nutrients provide high-energy electrons in the form of NADH, which are used by the protein complexes to pump protons from the matrix to the intermembrane space. This continuous pumping creates a proton gradient, where the positively charged protons are attracted to the more negative matrix. When the protons re-enter the matrix through the ATP synthase protein complex, they catalyze the production of ATP.
And then we have the cytoskeleton. That's on page 42. The cytoskeleton is the supporting framework of the cell. It determines the cell shape, strengthens the cell, it helps with the movement of the cell, and it may have extensions such as microvilli, cilia, or flagella. These are important to know. So the microvilli, these are folds of the cell membrane that greatly increase the surface area of a cell. These are generally found in areas that will help with absorbing nutrients, specifically the intestines. The microvilli are hair-like processes that are on the surface of the cell. They help move things in a wave-like direction. You're going to find this in the respiratory tract and also in the fallopian tubes, which is part of the female reproductive system. And then the flagella, these are similar to this cilia. They are found on sperm cells, and they do a whip-like motion to help the cell move. And that's all on the bottom of page 42. On page 43, we have movement through the cells. We have two types of movement. We have passive transport and we have active transport. Passive transport requires no energy, includes diffusion, osmosis, filtration, and facilitated diffusion. Active transport requires energy, includes transport by pumps and vesicles. Sorry.
Welcome to Movement Math and Cell Transportation. In this video, I'd like to talk about how cells move important objects into the cell and waste material out. Like all living things, cells must move objects into the cell and other objects out of the cell. There are two main ways a cell can accomplish this: passive or active transport. Let's first focus on passive transport. Passive transport does not require energy to move objects into and out of the cell. Think of passive transport like riding a bike down a hill. You don't have to pedal, you just coast. There are two main types of passive transport: diffusion and osmosis. In both of these, objects move from a high concentration to a low concentration. In this picture of the city, the first picture is high concentration, lots of people, and the second picture is of low concentration. Here are two examples of diffusion. Again, objects are moving from high concentration to low concentration. The air in the balloon quickly moved from the high concentration inside the balloon to the low concentration out of the balloon. Osmosis is simply the diffusion of water. Active transport requires energy. It's like riding a bike up a hill. We will look at three types. First, the cell uses a pump or a channel to move objects from a low concentration to a high concentration. This is like swimming upstream and requires energy. A second method is called endocytosis, in which the cell engulfs the object and brings it into the cell. Check out this picture. And finally, exocytosis is the opposite. The cell expels objects out of the cell. Hope this helps with cell transportation and active and passive transportation. Thanks for watching.
So on page 43, you have passive transport. The first type is diffusion. That's where particles move from an area of higher concentration to an area of lower concentration. So you have the highly concentrated dye tablet. You put it into the water, which is a solvent. The water dissolves into little particles. You can see the particles are evenly distributed here. So again, diffusion is when particles or solutes move from an area of higher concentration to an area of lower concentration. You have osmosis, which is the movement of water. If you want to think of the movement of water from higher concentration to lower concentration. If you look at the picture on page 43 on the bottom, you'll see the two beakers. The beaker on the left, it has two sides. The left side is 5% albumin. It has 6 particles in it and has more water. The right-hand side of the same beaker has 10% albumin. It has 12 particles and less water. So in the right-hand picture, you can see the water is moving from left to right. The water is moving where it's more concentration of water to where there's less water, to even out the percentage of albumin. So that is the movement of water, is osmosis from higher concentration to lower concentration. Osmolarity and tonicity. Okay, that has to do with how the cell is going to expand and shrink depending on the concentration of the solution that it is in. It says cells are essentially closed containers. Besides containing fluid, they also contain a variety of solutes, such as salts, sugar, acids, and bases. It's the concentration of these solutes in the fluid that determine whether and how much fluid moves into or out of the cell. So if you look at the 3 different pictures, you have an isotonic solution. The isotonic solution is where the solution is the same, the concentration of the solutes in the solution is the same as in the cells. So the amount of particles in the solution is the same as the cells. So the water moves evenly in and out of the red blood cell. Hypertonic solution. This contains a higher concentration of solutes within the fluid than compared to the red blood cell. So you can see that the water is moving from the red blood cell to the left outside of the cell. It's causing the cell to shrivel. The hypotonic solution contains a lower concentration of solutes compared with the fluid within the cell. This is going to cause the water to move into the cell. Okay, that is a hypotonic solution because there's more water on the outside of the cell. It's going to move in and the cell is going to swell or get bigger.
Welcome to Moo Moo Math and Science. In this video, I'd like to talk about types of solutions. And I'd like to talk about hypertonic, isotonic, and hypotonic. First, let's talk about a solution. A solution is a combination of the solvent and solute. The solvent is what dissolves, and the solute is what is being dissolved. In this cup of coffee, the water is the solvent, and the solute is the soluble parts of the coffee, the cream, and the sugar. Now, there's a relationship between the solute and the solvent. As the solute increases, the concentration of solvent decreases. And as the concentration of the solute decreases, the concentration of the solvent increases. So let's look at this blood cell. In hypertonic, it gets smaller or shrivels up. In isotonic, it stays the same. And in hypotonic, it gets larger. Let's see why. First, let's look at the hypertonic solution. This could be salt water. The solute increases, therefore the solvent concentration decreases. So water will flow from high to low, so there's a higher concentration of water inside the cell than outside the cell because the water is the solvent, so the water flows out, causing the cell to get smaller. In a hypotonic solution, such as distilled water, the solute decreases, therefore the concentration of the solvent increases. Water flows from high to low, so it flows from outside of the cell into the cell because there's a lower concentration in the cell. And finally, with an isotonic, it is just right. There is an equal concentration of water inside and out, so the cell remains the same. So there we go, 3 types of solutions: hyper, isotonic, and hypotonic. I hope this helps, and thanks for watching. Moo Moo Math uploads a new math and science video every day. Please subscribe.
Then on page 45, we reviewed facilitated diffusion. That is still a type of passive transport. A facilitated diffusion is when a solute needs, or a molecule needs, something to help move it through the plasma membrane. For example, a protein channel. So you can see in the picture, it says a solute such as glucose that can't pass through the plasma membrane enters a channel in a protein molecule that's part of the membrane. We talked about that when we talked about the plasma membrane, the protein channels. So you can see the protein, or the solute, fits into this protein channel, and it goes from higher concentration to lower concentration. So the protein channel is just facilitating that movement, facilitating that diffusion. Then we talk about active transport. Active transports are when solutes move up a concentration gradient. In other words, they move from an area that has less concentration to an area that has greater concentration. Since they're moving in this upward motion up the concentration gradient, they require energy to be able to do that. One of the most important examples of active transport is the sodium-potassium pump. If you see in the animation, you can see there's 4 different pictures. The first one is where you have the sodium that is connecting from the inside of the cell. They're the red molecules connecting into the receptor in the protein channel. Once all 3 are connected, you're seeing in 2, ATP is released to move them to the outside of the cell. The outside of the cell picks up the 2 potassium and brings the potassium into the cell. So that's the example of the sodium-potassium pump.
The sodium-potassium pump is a protein pump found in the cell membrane of all animal cells. Its main function is to transport sodium ions out of the cell and potassium ions into the cell. This serves a number of purposes, but is critically important to the function of neurons as it helps to maintain a higher concentration of sodium ions outside the cell and a higher concentration of potassium ions inside the cell. Preserving these differences in ion concentrations helps to stabilize the cell's membrane potential. This is critical for the neuron to be able to fire an action potential, which is the basis of the electrical signaling of neurons. The sodium-potassium pump is a large membrane-associated enzyme that uses the energy of adenosine triphosphate, or ATP, to transport sodium and potassium ions across the cell membrane. All of the details of how this is achieved are not yet fully clear, but the general process involves the binding of ATP to the pump, which promotes the binding of 3 sodium ions and the release of 2 previously bound potassium ions. ATP is broken down and transfers a chemical group known as a phosphate group to the pump, which prompts the pump to undergo a conformational change, or a change in its shape. This causes the bound sodium ions to be released into the extracellular fluid. At the same time, the pump binds 2 potassium ions, which also prompts another conformational change that returns the pump to its previous configuration, and the cycle begins again. Each cycle of the pump causes 3 sodium ions to move out of the cell and 2 potassium ions to move into the cell. Because there is one more positive charge leaving the cell than entering, there is a net loss of positive ions, which makes the resting membrane potential of the cell slightly more negative.
Okay, then you have transport into and out of the cell by vesicles. So that's on page 46. You have endocytosis and exocytosis. Endocytosis is the formation of the form of vesicular transport that brings substances into the cell. You have 2 types. You have phagocytosis, which is cell eating. So you can see it makes a vesicle around a particle, solid particle. Penocytosis is cell drinking. Okay, this occurs when the tiny vacuoles bring droplets of external fluid into the cell. Exocytosis is when substance or bring substances outside of the cell. So you can see in the diagram that you have substances within the vesicle within the cell, and they are ultimately fuses to the plasma membrane and releases that content outside of the cell. And that is the end of chapter 3. The test your knowledge questions are on page 54. Question 1, 2, 4, 5, 6, 8, and 9.